So our last speaker for the session is Leila. Leila's going to talk evidence for within-species transition between drought responses, strategies and events. So thank you very much.
Thank you. Hi, everyone. Today I'm going to talk about divergent drought response and strategies in Nicotiana Bethamiana. Plants growing in dry conditions can respond to water shortage by applying different strategies. Each of these strategies has its own features. In our lab, we have an Australian native plant, Nicotiana Bethamiana, which is globally used in a spectrum of research. It is predominantly growing in arid habitats across Australia. But none of its accessions show any obvious gazophytic morphological features. Therefore, we ask how such a tender-looking model plant could manage to survive hostile conditions and if its strategy is universal within the species. To answer this question, we impose different accessions of Bethamiana to drought stress and assess the rate of water loss from their detached leaves. We saw that there are such a dichotomy among the accessions of Bethamiana, especially those ones which are collected from arid locations such as CA from Central Australia, showed wilting phenotype sooner and lost water from its detached leaves earlier than or faster than the other ecotypes, especially those ones which are collected from less arid locations, such as NQ from northern Queensland. So we selected CA and NQ and look at their life cycle in response to drought stress. We noticed that once we impose a CA to drought stress, when it reached 5 to 6 leaf stage, it showed wilting phenotype after only 6 days and the permanent wilting phenotype after another 6 days. It means that after this time, plants couldn't be recovered from drought stress by recovering. These plants also set flower sooner than well-watered plants. For NQ plants, once it reached the same stage of plant development, we stopped the water supply. This plant showed wilting phenotype after 10 days, and after another 19 days, it showed permanent wilting phenotype. So totally, these plants could survive 29 days without receiving water supply. And also, this plant didn't set flowers unless it received water supply. So there are two major differences between these two accessions. One is wilting phenotype, the day of wilting phenotype, and the other one is permanent wilting phenotype. We look at these two accessions in more details. We look at the leaves of these accessions under the microscope. And we saw that after 60 minutes, once the majority of their stomata were closed, still CA leaves lost water more rapidly compared to NQ leaves. It means that something beyond their stomata is happening, which is the cuticle. So we measured cuticle conductance in UTAS, and we saw that for CA plants, no matter plants growing under well-water or water deficit conditions, there is no response for their cuticle conductance. But for NQ plants, not only under well-water conditions, their cuticle conductance is significantly lower than CA plants, but also in response to water shortage, they significantly reduce their cuticle conductance. So something happening in their cuticle. But why cuticle conductance is important? Because leaf transpiration is controlled by both stomata and cuticle, but during hot and dry conditions, plants first quickly close the stomata to reduce the amount of water loss. Therefore, cuticle takes the crucial role in preventing excessive amount of water loss. So it plays an important role for plant survival during hot and dry conditions. We look at the leaves of these plants in response to water shortage, and we saw that for NQ plants, we can see the wax crystals are larger and more abundant on the leaf surface compared to CA plants. Wax crystals are smaller and less abundant. Also, the thickness of cuticle increased significantly in NQ plants in response to water shortage, but no difference for CA plants. Therefore, something is happening in their cuticle. But what's that? Cuticle is composed of different components. Among them, alkanes, particularly very long-chain alkanes, have the greatest effect in determining water barrier properties of the cuticle, simply due to their hydrophobic nature. The substrates of alkanes are fatty acids. They are produced in the chloroplast, and then they are exported to the ER for the elongation cycles. But if plants require very long-chain fatty acids, they go through another cycle. In this cycle, SER2 plays the key role for the production of very long-chain fatty acids. The products of all these three cycles then converted to different chain lengths of alkanes by SER1 and SER3 proteins, and all these exported to the leaf surface to make the cuticle. So we look at them and we saw that for NQ plants, short chain alkanes and medium chain length alkanes increase in double in response to water shortage, but no difference in CA plants. And more importantly, we can see an 8-fold increase in the production of very long chain alkanes in NQ plants in response to water shortage, but we cannot see such a response in CA plants. And this happened 4 days before we can see the visible wilting phenotype in NQ plants. Then we measure gene expression. We look at the SER2, which is the key gene for the production of very long-chain fatty acids. We look at SER1 and SER2, which are responsible for producing alkanes, and LAX, which is responsible for exporting fatty acids from chloroplasts to the ER. The results show that all these genes substantially increased in NQ plants in response to water shortage, but there is little or no change in the expression of these genes for CA plants. Then we measure transition phase in UTAS. Why is important? Because once plants impose too hot conditions, there is a temperature at which cuticle lose its stability. Therefore, it's really important for plant survival during hot and dry conditions that usually happen together. We measured that and we saw that for CA plants, no matter plants growing under well water or water deficit conditions, it happened at 35 degrees. But for NQ plants, well water conditions such like CA, it happens at 35 degrees, but for water deficit conditions, it happened at 40 degrees. It means that those changes that happen in the cuticle structure of NQ not only makes the cuticle more impermeable to water loss, but also more stable to hot conditions, which are really important for plant survival. Then we look at the weather conditions of their climate, of their collection sites. And we saw that in both sites, approximately 80% of total rainfall happens during 4 months, which is wet season. The start of this wet season can be varied from year to year, but generally the pattern is the same. At CA collection site, the majority of rainfall happens during 2.5 months, and the rest of the year, the rain is very low, or the chance of getting second rain is pretty unlikely. And the temperature is above 40 degrees. So plants knows that it has 2.5 months to complete its life cycle. At NQ site, this rainfall was spread over 4 months and the temperature was below 40 degrees. So plants knows that it has 4 months to finish or complete its life cycle. Therefore, we end up with this model for their responses. CA plants, its response is well suited for its climate conditions. It's a scaper. It has 2.5 months to complete its life cycle, so it just keeps growing and it uses its leaf transpiration as a cooling system. For NQ plants, they have 4 months to complete life cycle, so it grows slowly. And if sudden drought happens during wet season, it evolved an ability to modify its cuticle structure, make a cuticle more impermeable to water loss, stay alive, because it knows that water will come back. But why we wanted to know these responses? Because we want to know what is the key regulators or genes behind such responses, different responses. We look at First, MIP transcription factors, because there are two MIPs which are reported in Arabidopsis, which are direct regulators of wax in response to drought, and another two indirect regulators in response to pathogens. But we didn't limit ourselves with these four MIPs. We look at all the MIPs which are reported in Arabidopsis to be responsive to environmental stresses. We selected all of them, blast them in bent genome, because we have a good assembly of both accessions, CA and NQ, and we did some phylogenetic analysis, and we end up with this tree. You can see in the clade, all the four MIP transcription factors from Arabidopsis cluster with 6 MIP genes from bent. There is also another MIP, MIP60, which is reported to be a stomatal regulator. Therefore, we have 6 MIP candidate genes, but we ask if these candidates are responsive to drought stress, so we measure their expression during drought experiment, 4 days before wilting days, 2 days, the day of wilting, and after wilting happened for both accessions, and for plants growing under both well water and water deficit conditions, and measure the expression of the key genes. You can see here a beautiful different pattern for their gene expression. At NQ, you can see the expression of MIP1 and 2 increase in response to drought, and also the expression of SER1 and SER2 increase. But for CA plants, the expression had no or little change in response to drought. Then we question, these MIPs are inducing in response to drought, but are they the regulators of wax biosynthesis genes? So we did transient assay for all these MIP genes, all the homologs for both accessions, and then measured the expression of the key genes. We saw that for MIP1, 2, and 6, they are able to positively upregulate the expression of wax genes, particularly SER2. And it is really interesting because it means that both accessions have MIPs that are capable of inducing SER2 gene, but in response to drought, only one of them induces the expression of SER2 gene. MIP 3, 4, 5 has little or no effect, or in some cases they act as a repressors. So we did some pull-down assays and we end up with this model. We are working on it to improve it, but this is our current model. We find that MIP 1 and 2, which are clustered with MIP 60, 96, and 94 from Arabidopsis, they act as positive regulators for alkane production. We have MIP 3, 4, 5, which are acting as negative regulators or repressors for alkane production. And this is interesting because it seems to be the first report of R2, R3 MIP transcription factors, which are acting as negative regulators for alkane production. And we have MIP 6, which clustered with a stomatal regulator, and our experiments show that it's a direct regulator of SER1 and SER2 genes. So we have somehow, in one hand, some positive regulators of alkane production, and the other hand, some negative regulators. And this is the plant who is making the decision to induce which MIP transcription factors when it comes to need. Therefore, by knowing these regulators and genes, we want to do some gene editing, which is our next step, our next goal, by using CRISPR-Cas technology. And you can know more about this technology by Boudini's and Sotomi's posters. Our first gene target is SER2, because as you remember, this gene was responsible for producing very long-chain fatty acids, which are then converted to very long-chain alkanes. So we selected that, and we look at the expression of this gene during drought stress for both accessions. We saw that in NQ, it's increased expression, but no change, even after wilting time for CA plants. Then we look at their sequence of their amino acids. There was a research showed that the first 100 amino acids are really important to determine the responsibility of SER2 gene. or protein. And we look at them, we saw that there is only one amino acid difference between these two accessions. Could it be the reason? We want to test it. We want to cut and paste this amino acid from NQ to CA and see if we can have a CA plant that are capable of producing very long-chain fatty acids. Because it's really important to have plants that are responsive to changing climate. Then this is our, oh, sorry. There should be another slide. Okay. One of my slides is, oh yes, it's here. Yes, it's here. Definitely, this is one target, which is, we are working on it, but definitely there are other genes that are interesting to follow and work on them, and that is the reason we are doing an RNA-seq experiment. Thank you for Center for providing us a fund to do this experiment. We are using 3 different accessions of Bethamiana because they are collected from different climatic conditions. They show different rates of water loss from their detached leaves. They show different days for wilting phenotype and permanent wilting phenotype. And we are doing cycling drought because we want to see if memory can help plant for a better, quicker, stronger response. And we are asking so many people around the center to help us to complete this project successfully. Then I want to say that, about the drought experiment, our paper just published recently, and if you want to know more about our experiments, you can have a look. And just today I received an email that it is also nominated as a Best International Research Award, which is really good, and I'm so happy for that, and thank you to all and the Center for providing us such opportunity. Thank you.
Congratulations.
Thank you.
Sorry. Any questions? I'm sure Peter's got a question. Sorry.
Maybe later. Peter always asks lots of questions.
Charlie, you have a question?
Yeah, I imagine the intent, beautiful biology. Wonderful biology. I imagine the intent is to improve crops.
Pardon?
The intent is to increase crops, improve crops.
Yes, I hope so.
So, fatty acids are really expensive to synthesize. Do you have a sense for how much ill penalty this trade may cost? Because you're synthesizing fatty acids.
For plants?
Yeah, for crops.
Peter is so interested. We
Sorry,
discussed this
Leila.
Yes.
So this is just the expanding leaf that actually changes in response to drought. So it's really clever. The plant doesn't change the cuticle in most of the plant. You get into drought and it takes the top leaves of the meristem and it protects that. Then it gets water and then it goes back to producing the normal cuticle and so on. So it's a temporary thing to respond to drought. It protects its sort of growing point, if you like, and a few leaves, and then it deprotects it when there's water around. Does that answer your question? So it's really quite clever. So it doesn't spend all its time sealing itself in and boiling in hot weather, as it were. It only does it when there's no water around, and it has to sort of hang on until the water comes. So I think that's quite clever of the plant. High IQ. So.
I think we better wind up because it's
Yeah,
afternoon
it's
tea time. Thank you
Thank
so
you.
much.
Thank you.
We are running a bit late for... We're still going to come back at 15:15? Yeah, we'll have to be back here at 3:15.